Hello and welcome to the movement disorder session of the Brain Conference. I'm Anne Rosser, I'm Professor of Clinical Neuroscience at Cardiff, and the co-host today is David Byrne, who's Professor of Movement Disorder Neurology in Newcastle. So we've got three fantastic expert speakers today who will provide some insights into the current state of the art for their particular areas of expertise. And we've got two really interesting data blitz presentations from young scientists. So we'd like to remind you to post your questions into the questions section, and we'll try to answer as many of them as we can. And at this stage, I'll hand over to David to introduce our first speaker.
Great, thanks very much, Anne, and good afternoon, everyone. Thanks for joining us. So it gives me great pleasure to introduce our first speaker, who I've known for quite a few years. It's Chris Kobylecki. He's an excellent clinical academic based in Manchester. We've seen Chris present in a number of fora before, including at the ABN. He's a clinician who sees huge numbers of patients with movement disorders and has a particular interest on atypical movement disorders. So that's the topic that he's going to speak to us on just now, which is on some top tips for the diagnosis of atypical Parkinsonian syndromes. So Chris, over to you, and thank you very much again.
Okay, thank you for the opportunity to speak today on the differential diagnosis of atypical Parkinsonian syndromes. So these are my disclosures. And the objectives for today are to really understand the process of differential diagnosis of these complex Parkinsonian disorders, to discuss current diagnostic criteria and put this in context with videos of relevant clinical signs, and to discuss the rational use of investigations. Now, we can divide atypical Parkinsonian syndromes up by their underlying pathology, for example, multiple system atrophy or dementia with Lewy bodies, by their underlying pathology, such as PSP and corticobasal degeneration. But it's also worth noting that there are numerous phenotypes that have been described resulting from underlying PSP pathology, like PSP Parkinsonism and gait freezing, which we'll go over later. The fact that there can be progressive aphasia overlapping with PSP and CBD pathology, and that frontal temporal dementia, behavioural and cognitive features are quite common in both of these syndromes. It's also worth noting that other pathologies can cause these syndromes, as well as that mimics of atypical Parkinsonian syndromes, which are often genetic, are important to be aware of, and we're going to talk a bit at the end of when to suspect those. So the first video here shows a 62-year-old man who presented to the clinic with progressive ataxia and early erectile dysfunction and urinary incontinence. And the sign that you're going to see on the video here is that on outstretching his arms, he has a significant myoclonic postural tremor, which is very different to what one sees in Parkinson's. As well as having presented with cerebellar features, he has evidence of Parkinsonism as well, and has obviously progressed rapidly because he's come in a wheelchair today. And his imaging shows a hot cross bun sign with a cruciform hyperintensity in the pons and atrophy in the cerebellum, consistent again with a diagnosis of MSA. So the diagnosis of MSA is made using the Gilman diagnostic criteria published in 2008. And for probable MSA, this requires a progressive autonomic failure with a relatively stringent definition, together with a poorly levered upper responsive Parkinsonian syndrome or cerebellar syndrome. And the criteria for possible MSA are less stringent in terms of their definition of autonomic failure, but require additional supportive features. And whilst these diagnostic criteria are currently being revised, I think they are a useful framework for thinking about the diagnosis of MSA. They also provide what are called red flags, which are indicators that the condition is likely to be MSA and not another condition like Parkinson's. And we've talked about myoclonic tremor already, and we'll talk about some of these other features listed in this table in the subsequent cases. It's also worth noting non-supportive features like prominent hallucinations, progressive dementia, a family history, as this disorder is usually sporadic, and a relatively older age of onset. So this video shows a 65-year-old lady who presented with a Parkinsonian syndrome with relatively early urinary incontinence and had brisk reflexes and extensor plantar responses. And over the course of a few months, she developed a marked forward flexion neck deformity, as shown here, consistent with anticolis. And this is relatively more common in MSA than in other conditions like Parkinson's, although it can be seen in people with Parkinson's. Incidentally, I'd always recommend that one checks for recent medication that's been started in Parkinson's, as dopamine agonists and amantadine are both reported to cause these axial deformities. And her level of anticolis is causing a disproportionate disability compared to the level of limb bradykinesia that she shows. The imaging here shows the lateral putamen slit sign with high signal in the lateral putamen consistent with vacuolation and gliosis and putaminal atrophy. And this can be used as a diagnostic feature in MSA. The next video shows a gentleman with a Parkinsonian syndrome who on levodopa developed marked abnormal movements of the orofacial musculature. And you can see abnormal dystonic type movements around the orofacial region, which were worse when he was speaking and eating, suggesting a degree of task specificity. And you'll see that despite being on levodopa, he had really very significant and severe bradykinesia of finger tapping and required a wheelchair to mobilize. And this orofacial dyskinesia was seen in around a quarter of people with pathologically confirmed MSA in a large clinico-pathological study by Gregor Werning and colleagues, and should be considered to be a red flag indicating the possible diagnosis of MSA. It's very different to the limb career-form dyskinesia that we see at peak dose in Parkinson's. I'm just going to play you this video to listen to in the first instance.
So can you just tell me what problems you're having right now? Okay. And what are the main problems? Are you having any problems with your swallowing at the moment? Okay. And I think, as you said, you've tried some treatments for it, but they caused dryness of the mouth, didn't they? Okay.
So this gentleman has a very harsh, high-pitched dysarthrophonia, which is very typical of what's seen in MSA and not so typical of, for example, Parkinson's. And the early development of severe speech and swallow problems, again, is an indicator of an atypical Parkinsonian syndrome. So PSP and MSA both developed swallow and speech problems at a much shorter latency compared to Parkinson's in a large clinico-pathological series. You'll see he has severe Parkinsonism and autonomic impairment as well. And the video also showed evidence of stridor during the day and sleep disordered breathing, which can include obstructive sleep apnea and stridor, are relatively common in MSA. It should be actively sought when we're considering this condition, as they are potentially treatable with modifications such as CPAP and tracheostomy. Early swallow problems obviously are relevant because they may require modification of diet or consideration of gastrostomy. So when we talk about levodopa responsiveness, it's worth noting that in this large prospective cohort study from a European cohort and replicated in the US cohort as well, up to a third of people with Parkinson's, with MSA, could have a reasonable response to levodopa, which could last for several years. And some people could also develop motor fluctuations and wearing off, more typically thought to be features of Parkinson's disease. In a recent clinico-pathological study for movement disorders, this again supported the idea that a reasonable proportion of people with MSA and PSP can have a moderate or marked response to levodopa. Obviously less common than one would see in Parkinson's. And this tells us two things, I think. One is that we shouldn't be too fixated on what the diagnostic criteria say that people have to be levodopa unresponsive, because that's clearly not the case from this published evidence. And secondly, that one shouldn't be nihilistic about the use of dopaminergic medications, particularly with significant limb Parkinsonism. And I'd urge everyone to have a trial of at least a gram of levodopa if tolerated to see whether this is beneficial, because it can be clinically relevant and helpful for people. There can be diagnostic difficulties, and this 63-year-old man was referred with a 5-year history of severe autonomic failure, gait and dexterity problems, and the referring team suspected him to have MSAP. But exploring his cognitive function in more detail revealed that he had fluctuating arousal and visual hallucinations with perceptuospatial impairment, and impaired executive function and significant global impairment of cognition, consistent with Lewy body disease. And his FDG PET showed significant posterior hypometabolism, again consistent with DLB. And this large clinico-pathological study from the Mayo Clinic indicated that DLB is the most common cause of MSA misdiagnosis. And so I would urge you to really explore the cognitive function, particularly visuospatial and hallucinations, in people referred with suspected MSA. And remember that autonomic failure plus Parkinsonism does not equal MSA. So it's very important to assess their cognitive function in particular. So this gentleman presented to the clinic with a Parkinsonian syndrome, and despite treatment with levodopa, remains quite bradykinetic. You also see that he has significantly reduced facial expression and frontalis overactivity.
Okay.
He also shows the applause sign, which was first reported in people with PSP, but is more likely to be a general feature of frontal subcortical disinhibition. On testing his eye movements, you'll see that his horizontal saccades, while slowed, are preserved in amplitude, but his vertical saccades are very markedly hypometric and slowed, consistent with a supranuclear gaze palsy. And so he has features which are consistent with progressive supranuclear palsy Parkinsonism. And to allow operationalization of this diagnosis, the Movement Disorder Society proposed recent changes to the diagnostic criteria for PSP, which didn't previously allow them to diagnose conditions other than Richardson syndrome. And they proposed a number of subtypes, including PSP with Parkinsonism, gait freezing, and frontotemporal dementia. Whilst most of these variant PSP syndromes may progress to a more diffuse Richardson syndrome phenotype, it is known that PSP Parkinsonism and gait freezing in particular appear to have slower disease progression and a slightly less aggressive disease course. And this is important for prognostication in these conditions, but also for designing clinical trials. And just to take you through the PSP diagnostic criteria, they consist of 4 domains of oculomotor dysfunction, postural instability, akinesia, cognitive disturbance, and 3 levels of diagnostic certainty. And putting his clinical features into this matrix, he has a vertical supranuclear gaze palsy and evidence of an akinetic rigid levodopa-resistant Parkinsonian syndrome. And together with additional clinical clues, he meets criteria for probable PSP with predominant Parkinsonism, allowing an earlier and more confident diagnosis of this subtype. This lady presented with a progressive gait disturbance without unprovoked falls, and on examination had slow vertical saccades without limb Parkinsonism. And her gait is somewhat unsteady when walking in a straight line, but you can see that when turning, she exhibits marked freezing of gait and then start hesitation. And so she has a slow velocity of vertical saccades and early progressive gait freezing within 3 years, which is consistent with the progressive gait freezing subtype of PSP, according to the new diagnostic criteria. And this is important, again, because patients may respond to some extent to interventions such as levodopa, and I'd urge a trial of levodopa in these patients, although sometimes the response is still not that great. Some ocular clues to the diagnosis of PSP include this gentleman who has features consistent with PSP Richardson syndrome, but on testing vertical saccades, you'll note that his eye movements follow a somewhat curvilinear trajectory or even diagonal trajectory. And this is consistent with the round the houses sign, where rather than being a straight up and down vertical saccadic eye movement, they take a more curvilinear trajectory. And this is thought to be people recruiting horizontal saccadic eye movements to produce a vertical saccade because their vertical saccades are so impaired. This lady who has progressive supranuclear palsy Richardson syndrome has difficulty with persistent eye closure and difficulty opening her eyes. And you'll see she's trying to employ a sensory trick here to open her eyes. And this is consistent with what's sometimes called apraxia of lid opening or pretarsal blepharospasm. In some cases, the term pretarsal blepharospasm may be preferred, as there's evidence for dystonic overactivity of these muscles in these patients, and they may respond to botulinum toxin injections, as indeed was the case here, as well as sensory tricks, including the use of ptosis props. So this is another important ocular clue to the diagnosis. Okay, so this lady presented to my clinic with a history of right-sided rest tremor, bradykinesia, and rigidity, initially consistent with the diagnosis of Parkinson's.
That's great.
But she didn't respond very well to levodopa, which prompted a re-evaluation of the clinical features.
Does that right arm ever do anything that you don't want it to?
No, it's just bone idle.
It's just bone idle, okay. Okay.
So she describes that her right arm just doesn't do anything and doesn't do what she wants it to, which is quite different to what we tend to hear from people with Parkinson's. And on testing bradykinesia, you'll see that she struggles to really even copy the movement that I'm doing with the right hand. And I felt this was consistent with a progressive apraxia. And indeed, her imaging shows asymmetric posterior frontal and parietal atrophy, more on the left side, consistent with a corticobasal syndrome, which is the diagnosis here. You'll see she's really struggling to copy non-representational hand postures with her hand. When I ask her to brush her teeth or mimic brushing her teeth, she also shows body part substitution and other evidences of apraxia.
Now with your right hand, can you show?
The next video shows a gentleman, again with the corticobasal syndrome, and he has evidence of myoclonus at rest, but also on performing actions. And he meets criteria for, according to the CBD diagnostic criteria for probable corticobasal syndrome, with limb rigidity and akinesia, limb dystonia, apraxia, a cortical sensory deficit, and evidence of myoclonus. And the myoclonus, as you'll see, is worse when he's doing actions here. And it's worth noting that corticobasal syndrome can be due to a number of underlying pathologies, and CBD pathology was only found to be the case in around a third of cases in this series from 2011. And it's also worth noting that corticobasal degeneration can present with various phenotypes, including the progressive speech disturbance similar to that we can see in people with PSP. When we talk about investigation of suspected atypical Parkinsonian syndromes, I would tend to go first to structural brain imaging with MRI. We know that dopaminergic imaging isn't really helpful to distinguish atypical Parkinsonism from Parkinson's disease or between the different Parkinsonian syndromes, although there is ongoing research on this. And this clinico-pathological study from Queen Square showed that the hummingbird sign, suggestive of midbrain atrophy, and the morning glory flower sign is also shown down here in panel C, has high specificity for PSP compared to MSA and healthy controls. The lateral putaminal slit sign of putaminal atrophy was not that helpful a diagnostic tool, although I still feel it's useful when you see it in somebody where you're suspecting MSA rather than an undifferentiated Parkinsonian syndrome. But the hot cross bun sign and middle cerebellar peduncle atrophy and high signal had high specificity for MSA compared to PSP in healthy controls. So these are useful rule-in investigations, if you like. They don't really help to rule out these conditions because of their relatively low sensitivity. The image on the left shows the pattern of FDG hypometabolism that's seen in atypical Parkinsonian conditions compared to Lewy body disorders, and this was from the same paper that showed that FDG PET was superior to dopaminergic imaging in the differential diagnosis of atypical Parkinsonism. The panel on the right just shows the typical pattern of hypometabolism of the basal ganglia in MSA compared to preserved metabolism in Parkinson's. As I said before, we see conditions that mimic these classical atypical Parkinsonian syndromes that are due to different underlying pathologies and often genetic conditions. And we also see that people present in an atypical manner that isn't usual for the classical atypical Parkinsonian syndromes. And this was reviewed very nicely in a paper by Maria Stamler and colleagues from Movement Disorders in 2013. But I've summarized the things that should make one think beyond MSA, PSP, and CBS. So, for example, an unusual age of onset, too young or too old, a strong family history, most of these disorders are sporadic, the additional neurological and non-neurological features are atypical, and the imaging suggests an alternative diagnosis. So the video on the left shows a gentleman with a tremulous Parkinsonian syndrome and early falls, and the video shows that he has significant slowing of vertical saccades. But he had a very strong family history with his mother, maternal uncle, and grandmother having died of frontotemporal dementia and significant frontotemporal atrophy. He had had a meningioma resected as well, hence the area of encephalomalacia. An investigation showed him to have a mutation in the microtubule-associated protein T gene, consistent with a genetic tauopathy. And this nice review of early onset progressive supranuclear palsy, by which the authors meant under the age of 55, in this review, they suggested testing for genes including MAPT and Niemann-Pick disease, amongst others, in young onset PSP cases.
Look at the camera.
The video on the right shows a gentleman with a relatively poorly levodopa responsive syndrome. And as you'll hear on the video, he has significant respiratory disturbance and was on non-invasive ventilation at this stage. His sister had a diagnosis of Parkinsonism and respiratory failure as well, and their mother had died at a young age of Parkinson's. And he had a mutation in the dynactin-1 gene consistent with Perry syndrome, an autosomal dominant condition where respiratory failure predominates. And somebody who's had very early mood disturbances or significant weight loss early in the course of their Parkinsonian syndrome, as well as a potential PSP syndrome, should also prompt thinking about Perry syndrome. The image at the top here shows a gentleman with a progressive cerebellar ataxia diagnosed elsewhere as MSA, but he had a very significant frontal behavioral disturbance, really out with what I'd normally expect to see in MSA. And the top panel shows this high signal within the middle cerebellar peduncles and global atrophy, particularly of the corpus callosum and cerebellum. And he had fragile X tremor ataxia syndrome confirmed on a genetic basis. The image below shows somebody who presented with a corticobasal syndrome but has a marked leukoencephalopathy on his imaging, very much not consistent with what we tend to see in corticobasal degeneration. There was a strong family history with an autosomal dominant flavor of an unspecified dementia. And he had pathological diagnosis confirmed of hereditary diffuse leukoencephalopathy with neuroaxonal spheroids, which is now diagnosed with a mutation in CSF1R gene. And this again is a genetic degenerative brain disease that can present with an atypical Parkinsonian disorder. Why does all this matter? Well, accurate diagnosis allows accurate prognostication and planning for the future. An accurate diagnosis allows identification of specific treatable complications, be that hypoventilation, Perry syndrome, or sleep disorder breathing in MSA. And in the future, where targeted treatments are available to the underlying molecular or genetic abnormalities, an exact diagnosis will be critical to allow disease-modifying therapy to potentially be used. So in conclusion, history and examination are critical in the differential diagnosis of atypical Parkinsonism. I'd ensure a proper trial of dopaminergic medication to ensure a lack of levodopa response, but also bearing in mind that some individuals may show a useful and beneficial levodopa response. Structural and functional imaging may provide useful diagnostic clues, and be aware of when to suspect rare conditions that can mimic the more classical atypical Parkinsonian syndromes. Thank you.
Fantastic, Chris. Well, thank you. That was a tour de force. You've crammed a huge amount into a very short space of time with some very illustrative videos there as well. So we're just waiting for 1 or 2 questions to pop in. So I'm going to take Chair's prerogative and just ask you 1 or 2 myself, if that's all right. We've got about 3 minutes actually, so not a lot of time. Chris, I mean, we're all aware of the sort of clinical pathological studies that sort of have highlighted the seminal work, for example, of Andrew Lee's, that highlighted the misdiagnosis rate, where patients had obviously very advanced had gone then and had the post-mortem correlation. In the current climate, in the current situation, knowing what we know now, do you have any sense as to the misdiagnosis rate currently for Parkinson's in, say, the first 3 years? What sort of numbers, what sort of percentages are we looking at?
Thanks, David. Yeah, I mean, I think that's in a way that's almost more relevant than what the misdiagnosis rate is at post-mortem, because it's very important for us to be able to get an accurate diagnosis. And I think there was some work that came out from the States a few years ago looking at misdiagnosis in early Parkinson's, and I think the misdiagnosis rate in early Parkinson's was significantly higher than perhaps we expect to see in the kind of brain bank studies. And I think in particular, PSP was quite highly represented in that early group, because we've seen people with PSP who've just looked like a sort of slightly suboptimally levodopa responsive Parkinsonian syndrome without any of the ocular signs or other clues. And I didn't have a time to play it today, but I've got a case example of just exactly that, of someone who was followed up over the years and eventually developed clear ocular signs. So I think the message that I would give to colleagues and junior colleagues who are training in movement disorders is, just keep examining your patients every time they come and keep an open mind. And if things aren't working out in the way that you think they should for the diagnosis that you've given them, then keep examining for those signs until you find them, until you come up with another answer. Because I think we all have to be prepared that we're going to be wrong some of the time in neurodegenerative conditions.
No, that's good advice, Chris. Well, one really quick question from me to finish with. Do you do an MRI scan on everybody presenting with suspected Parkinson's disease?
I don't actually, no, although I have seen there was a recent very good review of imaging in Parkinsonism and movement disorders clinical practice that suggested that perhaps one should. And I think with the advent of new markers like neuromelanin and things like that, that may well come in. I do it in anyone who's got any vaguely atypical sign, or in anyone in whom I suspect atypical Parkinsonism. They all get an MR if possible. But no, it's I think the threshold for doing them is coming down over the years rather than going up.
Well, it's nice to hear that you're still relying on your clinical acumen, which is considerable. So thanks very much indeed, Chris. Thanks a lot.
Thank you. Thanks very much.
So that's a super start to the session. And now we're going to move swiftly on to a data blitz session. And I'm absolutely delighted to introduce Tara Diviney, who actually trained as an undergrad in the University of Galway, an absolutely beautiful part of the world, I'm sure you all know. And she's now moved across to Oxford, and she's going to tell us about characterizing a novel GBA transgenic mouse model for Parkinson's disease. So Tara, over to you.
My name is Tara Diviney and I'm a PhD student at the University of Oxford. My research focuses on the characterization of a novel GBA-L444P BAC transgenic mouse model of Parkinson's disease. GBA encodes for the lysosomal enzyme glucocerebrosidase, or GCase. GCase is synthesized in the endoplasmic reticulum, where it's highly glycosylated. It loses its glycosylation upon transport to the lysosome, and in the lysosome, GCase catalyzes the conversion of glucosylceramide to glucose and ceramide. We are interested in GBA as heterozygous mutations in this gene are the strongest genetic risk factor for Parkinson's disease. Despite this, the pathological role of GBA in PD has not been fully elucidated, but two main theories reside. The first being that there is a reduction of GCase activity in the lysosome, which may induce accumulation of GCase substrates. And the second is that there is entrapment of mutant misfolded GCase in the ER, which can lead to ER stress. And so the loss of GCase activity and GCase retention in the ER could be broadly described as a loss of function and a gain of aberrant toxic function effect of the mutation. To further understand GBA's role in PD, our lab created a novel BAC transgenic humanized mouse model of GBA-associated PD expressing the human, either wild type or mutant, GBA transgene. Our crosses produced the genotypes in this table. First, non-transgenic mice have 2 copies of mouse wild-type GBA. Human wild-type mice have 1 copy of mouse wild-type GBA and 1 copy of the inserted wild-type human GBA transgene. Next, the L444P mutant mice have 1 copy of wild-type mouse GBA and 1 copy of the L444P human mutant transgene. And lastly, we have GBA hets, which have 1 copy of wild-type mouse GBA and so are heterozygous for wild-type mouse GBA. And I've included this table on subsequent slides to make the genotypes clearer. I mentioned that GBA mutations are associated with loss of GCase activity and gain of aberrant function due to GCase entrapment in the ER. And our mice recapitulate both of these pathological phenotypes. Firstly, GCase activity is reduced in the cortex of GBA hets and L444P mice. This reduction reaches significance at 18 months. Next, we see an abundance of the high molecular weight form of GCase in mutant mice. This high molecular weight form is the immature glycosylated form of GCase and is indicative of GCase retention in the ER. Following from this, fast-scan cyclic voltammetry experiments identified that L444P mice have a dopamine release deficit versus mice harboring the human wild-type GBA gene at 3 months. And this deficit was specific to dorsal striatum. Interestingly, GBA hets do not display a release deficit versus controls and versus L444Ps. As we see the deficit in L444P mice, but not in the GBA hets, this indicates that a gain of aberrant function effect of the L444P mutation and not loss of GCase activity alone is driving this deficit. Lastly, we found that the mice exhibit mild gait deficits. Although speed and cadence were similar across genotypes, the hind limb swing speed of L444P mice was significantly reduced. I would like to acknowledge my supervisors and other researchers who made this work possible. Please feel free to reach out to me or stop by my poster if you have any questions. Thank you.
Fantastic, Tara. Thank you very much. That really was a genuine data blitz. We have a couple of minutes, I think, for questions, if that's all right, Tara. I'm not sure if you're expecting any. And in fact, I think my co-chair, Anne, may well have a question for you, Anne.
Yeah, thank you, Tara. That was looks like a really interesting model. I just wondered, given that you've shown predominantly gain of function effects, I just wondered whether you're considering going on to produce a fully humanized mouse, maybe with homozygous GBA human mutation, and you might see a phenotype with that sort of mouse.
Yeah, so the reason why we're doing a heterozygous mutation in GBA is because homozygous mutations in GBA, which I didn't actually mention, they're a cause of Gaucher's disease, the lysosomal storage disorder. So it's the heterozygous GBA mutations that are the strongest risk factor for Parkinson's disease. So there have been models before with homozygous GBA mutations they say that it's kind of like a proxy for Parkinson's, but really they're modeling Gaucher's disease since that's caused by the homozygous mutation. So that's why we did the heterozygous mutation. And yeah, we tried to get 2 copies of the human gene in the mice. So say in our mutant mice, we have 1 copy of the human gene and then 1 copy of the wild type mouse gene. But we can't insert 2 copies of the human gene as they're embryonic lethal. So That's why the crosses are the way that they are.
Great answer, Tara. Are you going to be giving your mice cough mixture?
Actually, well, I've actually tried. I've administered Ambroxol to cells, which kind of a cough mixture, but not administered it to the mice yet. So I'm still looking through those results and gathering data on that at the moment. Yeah.
Fantastic, Tara. That's brilliant. Well, thank you very much, and well fielded on the questions as well. So I'm going to pass you all over now to Anne to introduce our next pair of speakers. Anne.
Okay, thank you, David. So it's a great pleasure now to introduce my colleague Lesley Jones, who I've known for well over 20 years. Lesley is Professor of Neuropsychiatric Genetics in Cardiff University. She's worked on key aspects of Huntington's disease pathogenesis for a long time, right back to the discovery of the Huntington gene in 1993. So Lesley's going to talk today about an area of Huntington's research that's attracting quite a lot of interest at the moment. So we look forward to hearing about Huntington's disease and expanding tail.
Hi, I'm Lesley Jones from Cardiff University. I'd like to thank the organisers for inviting me to speak at this very interesting meeting. And I'm going to talk to you about our work looking for Huntington's disease genetic modifiers. So I've been working on Huntington's disease since 1994, essentially, just after the gene and its mutation was discovered. So Huntington's disease, of course, was first described by George Huntington, who it's named after, although it certainly have been described in the literature before that. There are descriptions, but he actually noted its autosomal dominant inheritance, although that's not what he called it. And he noted its choreic movements and the psychiatric and behavioural symptoms that are often seen in this disease, and published this in 1872. And Huntington's causes a widespread neurodegeneration, but this starts focusing in the caudate and the putamen of the striatum. And during the disease, the death of the neurons causes this to go to a very tiny rim of tissue with a huge enlargement of the ventricle. And you can see that actually there's more widespread cell death, the cortical and interstices are enlarged, and the whole brain weighs less at death than a brain from a non-Huntington's person. It's caused, as most of you will know, by an expanded CAG repeat in the Huntington gene on the tip of the P arm of chromosome 4. There is a polymorphic CAG expansion, which in normal people, non-Huntington's patients, has up to 35 repeats. If it has more than 26 repeats, you may see expansion in the subsequent generations into the disease range. The disease is caused by 36 or more CAG repeats. It's in exon 1 of the Huntington gene, and it's coding. It forms a polyglutamine tract in the protein. The disease is usually diagnosed because of the motor symptoms, but there are actually changes that can be seen before this. So people's functional abilities will start to decline, and you'll start to see some erratic movements and cognitive decline. At the point of motor diagnosis, it's likely that in most cases around half of those medium spiny neurons in the striatum have already been lost. So the disease has quite a long prodrome. And if you express that expanded CAG repeat in a cell or an animal, any sort of model, what you see is that pretty well everything in that model will alter. And because of that, it's very hard to know what you should be addressing in order to treat the disease. So we took a view that we would go back to the genetics to see if we could see which targets we ought really to be addressing therapeutically. So the reason we went back to the genetics is because of this plot. So this is the registry data, and it's about 10,000 HD patients plotted here. And you can see there's a very strong correlation between the CAG length and the age at motor onset. However, what you can also see here is that for any one CAG length here, there's a really big variation in the age of onset of disease, and about half of that is heritable. So we know that it's caused by genetic variation, and our idea was that if we could identify that genetic variation, it would provide targets that might allow you to move people from here to here, so having onset much later, and that would be a big win. So we did a genome-wide association using age at motor onset in our GWAS, and essentially we took the age at which people were predicted to have onset from their CAG repeat length, and we looked at their actual age of onset, and we took this residual and used it as a quantitative trait in a genome-wide association. So we looked at the association with single nucleotide variants right across the genome, about 8 million of them, in a classic genome-wide association design. And this is our most recent study, which includes 5 waves of genotyping, or actually, yeah, 5 waves of genotyping. And you can see here, this is the genome-wide significant line, and you can see that we have a number of hits. And if you look at those hits, most of them are in or near genes that are involved in DNA repair. In fact, apart from FAN1 here, all of the other genes in red are involved in mismatch repair. There are a number of other genes that are also significant. We don't know their function, and we also saw a hit in Huntington itself, and this is not the CAG repeat length because we've excluded that from our analysis. So one of the things that made us think was that it was very likely that these modifiers were acting on the DNA, because these are DNA repair enzymes. You know, they cut DNA. MLH1 and FAN1 are both endonucleases. They can cleave the DNA. So knowing this, we looked at a series of other diseases that had a very similar mutational mechanism, so an expanded CAG repeat, mostly cerebellar ataxias, and we saw indeed that we got the same signal, implying that these modifiers modify the mutation, not the entire gene. We also did a study in collaboration with Sarah Tabrizi, where we tried to look at progression of disease rather than age of onset, because age of onset is not very it's not very exact. And when we did this, we used a data reduction technique, principal components analysis. So we put lots of our data together, and we developed a score that was taken 4 times in this prospective study. And what we found when we did this was that we saw a big hit at MSH3, and that had been implicated but wasn't genome-wide significant in our initial results. And each copy of the Myler allele at this locus gave 0.4 units a year increase in the total motor score and 0.1 unit a year decrease in the total functional capacity of the subjects. So we were very interested in this, and in fact it's been not replicated exactly, but looking at progression in a different way has also shown that the biggest hit is in MSH3 on chromosome 5. So we think that what we really should be interested in here is to go right back to the CAG repeat itself. Is DNA repair involved in Huntington's disease pathogenesis? Well, there is quite a bit of evidence. We see mutational instability, so the CAG repeat can change in length, and longer repeats tend to expand, and the longer they are, the more they expand. And this was first noted by Peggy Shelbourne and Anne Messer. And what was also noted is that if you delete some of the mismatch repair genes in mouse models, then you completely prevent this somatic expansion. And the somatic expansion that you see in human brain predicts onset of disease. So these are 2 patients with 47 repeats in the CAG tract, And these are histograms of 5 CAGs, rising in 5 CAG units here. And you can see that this person, who has a much more expanded repeat, had age of onset at 25, and this person with a less expanded repeat had age of onset at 40. And similarly for other repeats, it's a significant association. So what's going on? Why might somatic instability be happening? Well, we know that repeated DNA can adopt some unusual non-beta DNA structures. And those non-beta DNA structures of various types have single-stranded stretches. And those single-stranded stretches are susceptible to digestion by nucleases, essentially. So we think that these structures are seen by the DNA repair machinery as being lesions, and they try to correct them. And in doing this, they leave a single-stranded gap against which there has to be some gap-filling synthesis, and this causes both expansions and contractions, but in long repeats it tends to cause expansions. So we think there's a two-stage mechanism of pathogenesis. First of all, you have to have a repeat that's 36 or more CAGs long, and at that point the CAG repeat is licensed to expand, if you like, somatically, and it expands to a point where it is toxic to cells, and this threshold at which it's toxic is likely to be different in different cells. And what the downstream moiety is that causes disease is not clear, or there's been a huge amount of work done on the toxic protein, but nevertheless all of the other mechanisms based at the RNA level and the DNA level could also be taking place. So just to go back to DNA repair for a minute, and noting that all the proteins marked here in red are significant, have significant genes in RGUS. So the MUT-S dimers, which are either MUT-S, MSH2 and MSH3, or MSH2 and MSH6, they survey the DNA, and if they see something they think is a lesion, then they recruit the MUT-L dimer. And the MUT-L dimer is MLH1, PMS2, or one of these other dimers. So we see a significant signal in all of these dimers, and the MLH1 contains an endonuclease activity, so it can cut, so MUT-L can cut the DNA. The cleave section is removed, possibly by exo1, which has some significance in another study we've done, and the DNA is resynthesized by polymerase, we don't know which one is implied here, and ligated by Lig1, thus completing the repair of the so-called aberrant DNA. However, how do we take this and convert it to finding targets for therapies? So how do we attempt to find targets to move people from here to here? Well, we know that MSH3 knockout prevents somatic expansion. So these are Huntington's mice crossed with MSH3 knockout mice, heterozygous mice, and wild-type mice. And what you can see here is that each of these spikes is one additional CAG repeat long. This is a PCR reaction across the CAG repeat, and you get these hedgehog plots. And what you can see is that this extra expansion is completely ablated if you knock out MSH3. And actually, Bob LaHoo also showed that this was due to the ATPase domain of MSH3 because he knocked out that ATPase activity and it removed the somatic expansion. We also know that MLH1 knockdown reduces somatic expansion, interestingly, not just in CAG repeat disorders, but also in other repeat disorders caused by other sequences, so Fragile X and Friedreich's ataxia. FAN1 isn't part of the mismatch repair machinery, but it interacts with MLH1, and we know it protects against repeat expansion. This was first shown by Karen Usdin in a Fragile X mouse model, and when she knocked out FAN1, you can see that you are getting more repeats added here at 3 months and at 6 months compared with the wild-type mouse. So we think that what's happening here is you're getting somatic expansion. It probably occurs right from day one, if you like, perhaps from conception. That hasn't been established yet, but it's going on all the time. And this means that it's likely to be a mechanism that can be targeted before people get the disease, and certainly before they've lost at least quite a high proportion of those cells. We did some further work. We did some exome sequencing. So essentially, we wanted to look at people at the extreme, so people with very high or low age of onset compared with what their repeat length predicted. Because we thought that these people might be segregating variants of larger effect, so coding variants. And this is very useful for trying to work out mechanisms. This is available online. Work was done by Bran and Tom. The first thing we found was that the actual sequence of the CAG repeat makes a huge difference. So canonically, there's a CAA at the end of the CAG repeat tract with a CAG following. CAA also encodes glutamine. And if you have more CAAs, then you have much later onset. If you have no CAAs like this, then you have much earlier onset, and this is highly significant. It has been shown by others as well. And we looked at coding variants in various genes, and I'm just showing a couple here. So these red arrows are people with earlier onset, and the green arrows people with later onset, and you can see these earlier onset arrows variants cluster in two domains. So this is the nuclease domain of FAN1, and this is the DNA binding domain. You can see there's a significant excess of damaging and loss of function variants in FAN1 early onset people. The segregation isn't quite so striking for MSH3, and it's not significant, but it's the other way around. So it implies that you're seeing the opposite effect. So Tom made a series of recombinant proteins carrying some of these variants and used them in nuclease assays. So the first shot here, panel A, is some lymphoblastoid cells carrying the R507H heterozygotes, and they were used in a mitomycin C assay. So FAN1 is known to cause to resolve interstrand crosslinks, and mitomycin C causes interstrand crosslinks. If they're not resolved, the cells don't grow, and you can see here that the cells aren't growing when they have this mutation in them compared with the wild type. These are lymphoblastoid cells from the patients. And Tom also did an assay here where with a 5' flap, which is FAN1's preferred substrate, and he used a number of variants, and you can see that the wild type produces a nice product here, and the control nuclease dead variant produces no product. R507H also produces no product. So these are plotted here, and interestingly, if you plot them against the residual age at motor onset, this gives you a significant correlation. So we think the nuclease activity is really important in FAN1 is really important in determining the age of onset of Huntington's disease. We also did some modelling in iPS cells to follow this up. Jasmine Donaldson did this as part of her PhD. She knocked FAN1 out, and she also made a nuclease deficient variant. The D960A has no nuclease activity. And you can see here in these hedgehog plots that if you knock FAN1 out here, or if you have a D960A nuclease dead homozygote, after 42 days in culture, these iPSC lines show more expansion in both cases. And this is plotted here. So I direct you to this. This is looking at the modal CAG repeat length. And you can see here that if you knock out FAN1 in yellow, or you have a homozygote D960A mutation in blue, that you get identical results. If you have a heterozygote nuclease dead knockout, you get something in between, so it looks additive, and this is the wild type here. And we know that FAM1 nuclease activity and interaction with MLH1 both promote expansion, because there were 2 papers last year published showing these events. And this is interesting, partly because the DNA repair pathways are involved in many cancers, as you will know. And these are the double-stranded break repair mechanisms and the single-stranded break repair mechanisms. They have a lot of overlapping proteins within the different types of repair, which I'm not going to go into. But the important thing I'd like you to pay attention to here really is that these proteins in red here are proteins to which there are already therapeutic design going on. So it's possible that we can tap into the oncological space and use some of these both to probe mechanism and possibly to try and generate therapeutics for Huntington's and other repeat disorders as well. So what we think is going on is, so we know that Huntington with an expanded CAG repeat causes increased DNA damage in cells and in mice. And we think if you get that increased DNA damage, that itself starts to cause more expansion of the CAG repeat. So then you get more DNA damage occurring and you induce the DNA damage machinery and you get yet more expansion of the repeat. So you end up with this toxic, vicious cycle where you've got increased repair causing increased somatic expansion and going on to cause more repair. So we think that these studies are helping us to move into the translational space for Huntington's disease to try and prevent that somatic expansion that underlies modification of the age of onset of disease in patients. So I'd like to thank hugely the patients, families, clinicians and researchers who made these studies possible by spending many years collecting patient cohorts and the clinical information to enable us to do our studies. And I'd like to thank my group pictured here a couple of years ago when we were allowed to get together and my collaborators. And thank you very much for listening.
So thank you very much, Lesley. That was a lovely presentation and really exciting work. So I just wanted to ask, the evidence here is really pushing us towards the conclusion that DNA repair machinery is central to well, not just HD pathogenesis, but pathogenesis in triplet repeat diseases. But of course, manipulating DNA repair machinery raises anxieties about inducing malignancies. So I wondered whether you'd like to comment on that.
So I think you have to pick your targets carefully, but it's very clear, and clearly people who are generating therapeutics in the oncological space are looking to do something different because they are looking to prevent cell division, essentially, to make cells die. We want to keep the cells alive, and we don't want to induce any carcinogenic effects. So I think there are some targets which would allow you to intervene in this pathway without necessarily having really widespread effects on the DNA repair machinery. So for instance, PMS2 and MLH1, which are both underlie Lynch syndrome, you certainly would not want to use in this sort of strategy. But I think there are other targets. MSH3, for instance, has attracted a lot of attention that could be used as targets that would allow you to develop therapeutics. And I think the other thing to be said is that you wouldn't want to knock them out completely, but that likely, the mouse data probably say that you wouldn't necessarily have to do that.
Okay, thanks very much, Lesley. Really exciting science. I'm afraid for the interest of time, we're going to have to move on, but I'm sure there'll be questions for you in the chat to answer later. So thank you. So we'll now move on to the next data blitz, which is by Chun Chen. from the Wellcome Centre for Mitochondrial Research at Newcastle University, and she's going to talk about imaging mass cytometry analysis of mitochondrial defects in human substantia nigra neurons in Parkinson's disease.
Mitochondrial dysfunction has been heavily implicated in Parkinson's pathogenesis, in particular the loss of vulnerable dopaminergic neurons in the SN. Here we utilized imaging mass cytometry, short for IMC, to perform complex profiling of mitochondrial protein expression in midbrain section, using antibodies conjugated with rare earth heavy metals. Difference to fluorescent imaging techniques, in which the number of targeted proteins is limited due to the wavelength of fluorophores. IMC detects the abundance of different metal tags, which reflects the abundance of targeted proteins in the tissue. These systems allows up to 47 different proteins to be measured simultaneously at the resolution of single neurons. The example images here showing the IMC detections of up to 11 mitochondrial proteins in individual dopaminergic neuron. Our first IMC experiment analyzed in detail the expression level of mitochondrial oxphorus proteins. From this data set, we identified a widespread decrease involving all 5 oxphorus subunits in Parkinson's neurons, despite heterogeneities between individuals. This dataset also allows us to investigate correlation between proteins in pathways. For example, the change in the relationship between nuclear and mitochondrial DNA encoded subunit between Parkinson's and control neurons. With an astrocytic cell marker, GFAP, added into our oxphorus panel, we also identified deficiencies in oxphorus complexes proteins within the astrocytes of the SN in Parkinson's, suggesting the supportive role of this cell population towards neuronal survival may be affected. To further understand whether impaired mitochondrial proteostasis is present in the Parkinson's SN and is contribute to Parkinson's-related neuronal loss, our ongoing IMC study provides a larger panel of proteins involving in the mitochondrial quality control system. We have found significant decreases in signaling factors that regulate mitochondrial protein synthesis, mitochondrial proteases, and mitophagy in PD. Meanwhile, increase of mitochondria and phosphorylated ubiquitins were observed in PD neurons with alpha-synuclein aggregation compared to those without pathology. Overall, our studies highlight the use of IMC for the assessment of mitochondrial protein expression in the SN, providing post-mortem evidence to support the complexity of impaired mitochondrial protein homeostasis in Parkinson's, which may increase the neuronal vulnerability to age-related oxidative stress, leading to early neural degeneration. Thank you.
Thank you, Chun. They were beautiful images and clearly a really powerful technology. Could I ask, you mentioned that you see differences in the mitochondrial phosphorylated ubiquitins in individual neurons, depending on their alpha-synuclein aggregate status. So do you see any other differences in other mitochondrial markers between those, depending on the aggregate status?
I would say that's quite we didn't expect to see that, but when we looked through the image, we found that actually in PD neurons with alpha-synuclein aggregates, they also show that aggregations of phosphorylated ubiquitin alongside an increased expression of some mitochondrial proteases like LONP and HETCH2. So we think it might be linked to the UPS systems being interrupted, which kind of link between mitochondrial dysfunction and pathogenic protein aggregation lysosome into the PD-related neural degeneration. Yeah.
Okay, and have you looked at non-TH neurons in your sections as well as TH neurons?
We did. We actually discussed with Jim from Chicago Jim somewhere. I'm not sure I pronounced her name right. And he suggests us looking at that as well. And we did do this analysis. We didn't really find a very big difference, but also at the same time, we don't really know those neurons in the SN, they don't show TH positive, whether they lost functions, they are like pre-active or they just another type of neural population there. But it's definitely something we are going to look deeper into.
Okay, thank you. That's really interesting. I'm afraid we'll have to move on to the next talk now.
Thanks, Anne. So anybody who's thinking of going to get a cup of tea or an early finish, don't, because we're finishing an absolute high here with another cracking talk. This one is from Michele Hugh, who's professor in Oxford and another outstanding clinical academic from the UK, who basically got the oil tanker moving, that is the Oxford Discovery Project from the clinical perspective. And Michele, I'm sure, is about to tell us about work in relationship to that juggernaut of a project. And so make sure that you've plenty of questions for Michele ready at the end. Over to you, Michele.
Many thanks to the organisers for giving me the opportunity today to tell you a little bit about managing Parkinson's and the translation from research to clinical practice. I'm going to cover in my talk 3 things which will hopefully give you some top tips. Firstly, how to get the diagnosis right. Secondly, the role of exercise in Parkinson's. And lastly, the importance of sleep and anxiety management to optimize motor symptoms. Getting the diagnosis right from the clinician perspective is generally involves a comparison between clinical diagnostic accuracy versus neuropathological diagnosis. And in this recently published study from the ASAND cohort in Arizona, in participants with a high rate of brain donation, 2 clinical diagnoses were made. Firstly, possible Parkinson's, and secondly, probable Parkinson's, using the presence of 2 out of 3 cardinal clinical signs for Parkinson's and the presence or absence of benefit with dopamine, levodopa. The bottom line from this study is that we're not very good, even now, at making a clinical diagnosis of Parkinson's. At best, we have an overall 89% accuracy, and our sensitivity and specificity at first visit goes down to 83 to 86%. So diagnostic accuracy for Parkinson's tends to be best if there is at least 5 years of follow-up, if an individual develops motor fluctuations and has hyposmia. However, diagnostic accuracy tends to be worse in the elderly, in those who are not treated or have a poor benefit to levodopa, as we would expect. So this does raise the caveat that interpreting results of cohort studies, particularly in those with early Parkinson's, needs to be done with care. From a patient perspective, getting the diagnosis right is a very different question to what the clinician has. And Peter Harling, a patient of mine with Parkinson's, wrote this paper in 2015. And what it covers is the emotional impact of being diagnosed with Parkinson's on the individual, which is very painful. And Peter then described the 3 phases of emotion an individual will go through following their diagnosis, including shock, disbelief, anger and frustration, then rationalization, followed by, hopefully, depression and acceptance. But we do see many patients, unfortunately, who will get stuck at, say, phase 4 depression without moving on to acceptance, which will then allow them to proactively manage symptoms. So in response to these difficulties, the First Steps peer-led program was developed as a concept by people with Parkinson's in Oxfordshire, including Peter. And this was then piloted in 2016. And based on the success, it was then nationalised to an online First Steps programme, which I want to publicise today because of its benefit. So in addition to just anecdotal individual reports shown here, as a research group working with Professor Helen Dawes at Brookes University, we have tried to gather more evidence on the benefit of First Steps. We compared 18 people with Parkinson's who had gone through the First Steps course to 22 recently diagnosed people with Parkinson's in the active control group receiving best current management. A single semi-structured interview was used, and a significant improvement in exercise behaviour and coping mechanisms was seen following the First Steps intervention. So this study, for the first time, provides evidence of the impact of peer-led psychosocial interventions, including First Steps, and I would recommend it as a tool to help individuals at diagnosis and following. Importantly, this intervention is led entirely by people with Parkinson's themselves, rather than allied health professionals or clinicians. So in summary, getting the diagnosis right involves different understandings from both the clinician and the patient. And it's important for us as clinicians to really understand the emotional impact of a diagnosis, to give the person time to elicit their symptoms and to establish a relationship and rapport, because you need to manage them going forward, and things are unfortunately going to progress in terms of their symptoms. A discussion about the implications of the diagnosis and the various treatment options is needed, and referral to the Parkinson's nurse team, provided you have one, is key. We have put together an information pack for newly diagnosed, which signposts local services, First Steps and other courses, exercise, Parkinson's UK and other charities, and the DVLA guidelines. It's important that you document and provide written as well as oral information on the different treatment side effects. These include impulsive compulsive disorder, excessive daytime somnolence, and psychosis. And NICE guidelines state really very clearly that at diagnosis, that individual needs access to ongoing clinical monitoring, medicines adjustments, and a continuing point of contact that could be ideally provided with a Parkinson nurse working with a clinician. Moving on to the role of exercise in Parkinson's, there is growing evidence of benefit for exercise on both motor and non-motor symptoms. This large multi-center phase 2 randomized controlled trial called SPARKS was US-based. It recruited early untreated people with Parkinson's at Hun and Yar 1 to 2, who were expected to be able to continue for 6 months without medication. Individuals were randomized to high or moderate intensity treadmill exercise or usual care, and they were asked to exercise for 45 minutes on the treadmill 3 days a week, both at home and on site with heart rate monitoring. The primary outcome was a change at 6 months in the untreated or overnight off withdrawn UPDRS motor score for individuals who had to go on to levodopa or other therapies. The key findings are that of the 128 people randomized, there was a significant reduction in the UPDRS off motor score in those in the high intensity group compared to the usual care group. And this was just significant. There was, however, no benefit in the moderate intensity group, and around 30% of people had adverse events related to exercise, such as falls or mild injury. In a more recent study published by Bas Bloem's group in Holland, a single-centre, double-blind phase 2 randomised trial called Parkinshape was performed, and this looked at people on treatment with early disease with a Hernian 2 or lower. People were randomised 1-to-1 to aerobic intervention using either a home trainer, which was stationary at home, or stretching with an active control group. So this did provide a degree of blinding. They were asked to exercise for 30 to 45 minutes, 3 times a week at home over 6 months, and they had remote supervision, but they improved compliance with a motivational app and virtual reality gamification. Physical fitness was assessed with VO2, and this study found a clear benefit of aerobic exercise in the off UPDRS motor score, with a reduction of 4.2 points on the UPDRS motor scale in favour of exercise. Around 6 to 11% of people had adverse events, including falls, knee injuries, and syncopal faints. But there was a weak but significant correlation between the improved motor scores and increased physical fitness. Can exercise performance be enhanced other than the methods I've just described? In Oxford, we worked with Professor Kieran Clark, who developed a ketone ester drink originally designed to improve performance in combatants in the US military. However, here we're giving it to people with Parkinson's, 14 in numbers, and we're measuring their exercise performance on a stationary bike with a warm-up followed by cycling against resistance at 80 RPM. We're measuring blood levels of the ketone, the beta-D-hydroxybutyrate, given with the carbohydrate or in a blinded way with a carbohydrate supplement alone. And then, as you can see here in the yellow bars, which show the effect of delta ketone with the carbohydrate drink versus the blue, which is carbohydrate alone, there's a significant improvement in exercise performance with ability to continue cycling at 80 RPM. And this varies from individual to individual, but overall, there's around a 25% improvement. So this study does suggest that by supplementing with ketone ester, you can improve endurance exercise and potentially the aerobic benefit of exercise. We now need to do more studies in larger number of patients and over a longer time period to assess the benefit. So to summarize about the role of exercise in Parkinson's, there's growing human evidence in addition to the animal studies that high intensity aerobic exercise benefits motoric symptoms in Parkinson's. The correlation between the improved physical fitness and the motor off scores suggests this is a genuine effect of aerobic exercise, and it's unlikely to be placebo effect related due to controlled trials. There are risks which are not insignificant, but in general, these are minor side effects. The mechanisms are unclear, but it is likely to be all of these things, include improved dopamine release, neurotropic factors, angiogenesis, immune system, mitochondrial function, leading to improved neuroplasticity. Lastly, what about the role and the interaction of sleep and anxiety on motor functioning? And in this regard, how has Parkinson phenotyping research actually changed what I do in the clinic? So in this study, which I've presented before, we worked with Donald Gossett, professor and PI of the Tracking UK cohort, very similar early PD cohort, combining with Discovery to get over 2,500 people who were studied at baseline and longitudinally over 3 years. So here we're using clinical information across a range of motor, non-motor, cognitive domains in a data-driven approach to define PD clusters using k-means cluster analysis. And in this graph, the main take-home finding in this respect is cluster 3, present in 21% of all of the early PD patients. Now, this cluster show worse non-tremor motor disease, worse sleep and psychological well-being that tend to cluster together. So what does this really mean? Well, if we start to drill down onto the specifics of the factors, we can see here that factor 1, which is psychological well-being, comprises a lot of elements that patients commonly complain of: apathy, fatigue, pain, neuroticism on the BFI, anxiety, depression. This is also linked to having a non-tremor motor phenotype, or PIGD, on MDS-UPDRS sub-items. And it's lastly linked to worse sleep in terms of higher scores on the RBD screening questionnaire and increased daytime somnolence. Many patients I see in clinic describe this complex interplay between anxiety and that worsening of their motor symptoms. So as soon as they get anxious, their tremor gets worse, they get bradykinesia, they may freeze, they find movement initiation difficult, this person describing walking in treacle. And the problem is that on the remote telephone call that you're assessing the patient, or even on a video, it can be impossible to actually distinguish generalized anxiety with acute exacerbation versus motoric wearing off symptoms that are primarily driven by motor or dopamine deficit. So having gone through this many times with patients, I have tended to approach this using this kind of cluster data by firstly screening for anxiety and depression as concomitant with an individual's Parkinson's. So that gives you a baseline idea of is it present and how severe. I'm benefiting from regular psychiatry input with my consultant psychiatry colleague Ivan Koychev with a monthly PDMD-PT meeting where we might discuss cases. And then it is really down to whether you think this is primarily anxiety driven, in which case you might go down the SSRI, SNRI or pregabalin route, or if you're not sure, the lorazepam test. Now, this involves giving somebody a low dose of lorazepam, increasing gradually over a week, and then seeing what actually improves. So if with lorazepam, the anxiety and secondary motor worsening gets better, you know that you don't need to actually review their dopaminergic therapy, but maybe transition to long-term anxiety management using the things that I've mentioned. However, if the anxiety is good and with the lorazepam we don't see any improvement, then it's probably going to be looking for wearing off motorically and adjusting their levodopa interval or adding in a secondary agent. Sleep really matters in Parkinson's, and in addition to the episodes of RBD seen here on the video, we see accelerated normal aging in people with Parkinson's, giving a range of complex problems. However, we know that specifically RBD increases motor, cognitive and mood progression in people newly diagnosed with Parkinson's. And this study from my group looked at 923 people with Parkinson's from the Oxford Discovery cohort, followed up over 5 years, and dichotomized into those with and without probable RBD on a screening questionnaire. So we see a third of early diagnosed PD patients have RBD at diagnosis and continue to have it over the next 5 years, and that having RBD means faster progression in motor, mood and cognitive symptoms, defining a more aggressive subtype. And these are just some of the plots comparing people with RBD in orange versus those without in green, showing faster decline in motor, non-motor and also cognitive scores. So this raised the question of whether treating RBD and sleep disturbance might be neuroprotective for Parkinson's. And this peer-reviewed paper, which is out in bioarchives, suggested that treatment with melatonin through a chronobiotic regime may actually reduce PD phenoconversion in RBD. However, this is untested or replicated in other cohorts and is provisional data only. Treatment of RBD is complex, and I've summarized it here for the purposes of time. So just to summarize and to end, my top tips for managing Parkinson's include getting the diagnosis right, both from a clinician and a patient perspective, emphasizing the importance of exercise, particularly early on at diagnosis. It enables the patient to have a positive approach, proactive management of their symptoms, and it may be neuroprotective, as is management potentially of sleep and anxiety, which reduce that vicious motor anxiety loop and might in the longer term neuroprotect patients in its own right. Thank you.
Great, thanks, Michele. That's fantastic. You covered a huge amount there in a short time. We've only got a couple of minutes now till the end, but we've had a question in from Graham Warner, who's asking, how is the Parkinson's disease anxiety loop unique, say, compared with multiple sclerosis or even long COVID?
Yeah, I think that's a great question. And we're all seeing, of course, physical symptoms being worsened, of course, during COVID due to anxiety and depression. I think the key difference in Parkinson's is the underlying pathogenesis and the neurotransmitter deficits that we see. So, of course, we see striatal dopamine reduction, but we also have shown reductions in serotonin localizing to the raphe nucleus, again, metabolized through that whole dopamine pathway that are probably driving anxiety and potentially altered reward through this sort of limbic ventral striatal system. So I think that there's a particular reason why many patients have prodromal anxiety often predating their motor onset symptoms by 10, possibly even 15 years, and that they then have the dopamine. But I think that the two interplay really in a sort of crescendoing cycle that for me I see just so commonly, and it really contributes to difficulty management.
Thanks, Michele. Brilliant, great answer, and a great question, Graham. Thank you. So we're needing to draw this to a whole, and we've had a fantastic number of colleagues join us, well over the 210 mark. So thanks to you all for your time. It just remains for me to thank all of the speakers for their wonderful contributions. I'm sure you've enjoyed all the talks. I know I certainly have. And my co-chair, Anne Rosser, who has done a fabulous job in choreographing this whole session. So thanks, Anne. And to, of course, guarantors of Brain for their support for this whole meeting. Thank you all.